Wolfgang Parak now. He's from the Department of Physics at the Marburg University and Biomagune San Sebastian. Some physicochemical aspects that may complicate active targeting.
Thank you. Good, thank you very much. So I just realized I have too much hair, so it's complicated to get that headphone on. But anyway, now it works. So I would like to talk to you a little bit about targeting of nanoparticles. How do I switch the slides? Who's that one? Okay, here. Good. So, and in particular, I would like to talk a little bit about active versus passive targeting. So if you read about active targeting in the literature, what people think they want to do, you have, for example, a tumor, and this tumor, you have some receptors overexpressed, for example, here. So the idea is just to bind ligand molecules to the surface of a nanoparticle, to inject it, and then this nanoparticle would go here to the tumor, and given by these ligands, it would bind to the receptors on the surface of the tumor. But we all know from literature that this one sometimes does not work. And what I would like to do with you now, I would like to discuss 3 different possibilities why this active targeting might go wrong. So, I want to do it from more from a particle standpoint than from a, let's say, medical standpoint. So let's imagine we have here our particle. So this one could be here, for example, gold for photothermal therapy, or it could be a magnetic particle for hyperthermia. You always have a coating around. And then, as I mentioned, we have some ligands, for example, antibodies attached to do the targeting. But that's the particle in the test tube. And the story I want to make is that this particle in the test tube is not the particle what you at the end will have in the body. So if this particle is already injected in the blood, you will get absorption of proteins. This one might camouflage partly the ligand surface. Then you can have degradation. For example, enzymes in the body, they may degrade the ligand shell. And of course, if the ligand shell and the antibody comes off, then basically your targeting capability would be lost. And what's also important here, if you compare from here to here, you have less ligand than you have here. So these are all things what can happen. And about this degradation part, I talked in the last CLINAM. So I want to focus on 2 other parts. At the end, I will tell a little bit about size measurements, how we could measure, let's say, this protein corona formation potentially in vivo. And in the first part, I just want to discuss a possibility. So if basically some of the ligand may come off, what is the importance at any rate for ligand density? So is it good to have a particle with a lot of ligands, or is it better to have a particle with less ligands? So in order to go ahead, first we have to think about the material, and we wanted to make precisely controlled particles. So to have particles with an exactly controlled number of ligands. So now from the chemistry we know it doesn't work. So if we have, let's say, particles and we have ligands, and we even mix them in the ratio, for example, 1:1, we would also get some particles with no ligand, some particles with 1 ligand, and some particles with 2 ligand. You always will get a distribution. But we wanted to make it more precise, so we thought of a way of fractionating the particles to get really particles with a defined density of ligands. So the way how you can do this is using gel electrophoresis. A gel is a porous matrix, and if you load particles, for example, here in if you load particles, for example, here. So these particles are negatively charged, and in the gel they migrate towards the plus pole. Now the gel is a porous matrix, so these particles have to squeeze through the pores, and what effectively you're doing, you're having a filter for size. So I have to mention the charge density, of course, also plays a role. If there's no charge, there's no electrophoretic mobility. But if you have, let's say, conjugates where your charge density more or less remains constant, what you're effectively doing is sorting by size. So this one we started long ago. So it was actually when I was in 2000 postdoc in Berkeley with the Alevisatos group. So just to show you, here's this example. If you have gold and you add some oligonucleotide and you mix it in different ratio, you can separate different bands on the gel. So what's obvious, if you add more and more of this DNA, everything becomes bigger and bigger, so the particles migrate slower on the gel because they can go harder through the pores. Now, if you can see here, this one is a band of free gold, but if you add more and more DNA, you can see you get an onset of different bands. And the first band we ascribe to gold with just 1 DNA, gold with just 2 DNA, gold with just 3 DNA, and so on. So in this way, we really can fractionate particles with a precisely controlled amount of ligands per particle. Now, you want to ask for controls. Here's one good control. If you do this one here for one type of oligonucleotide, you do the second one for the second type of oligonucleotide, you mix them together and you're getting dimers. If you had, for example, here a second strand of DNA, you would bind also a gold particle here, and instead of dimers, you would get trimers. So we did a lot of control, and with the structures, you can really make with quite high affinity. Now, this one was 15 years ago, and that was, of course, the most easy system because this sialated DNA you can readily link to the surface of gold. But we had to make another step to be more general because, of course, for all this electrophoretic separation, you need particles with a very narrow size distribution. And we wanted to apply it for all different types of particles. Now, as I mentioned, for gold, it's quite easy to link something, but if we want to go, for example, to superparamagnetic particles or to quantum dots, you need a different way. Now, we synthesize these particles in organic solvent, but at the end, we need, of course, to have them in water. So what we developed is a polymer coating technique. You can see here, this one would be the particle kept by hydrophobic surfactant molecules, but that's an organic phase. Now, if you take an amphiphilic polymer, you can simply add this amphiphilic polymer in the organic phase, driven by hydrophobic interaction. It will wrap around the particle. Now you evaporate the solvent, and because now this backbone is polar, you can disperse all these particles easily in water. Now, the nice thing is this works very general for all types of particles, and it gives you a very narrow size distribution. Now, we had to go for a more general linker, and one general linker is polyethylene glycol. So remember, our idea is if the particle becomes bigger with each ligand, we can just separate by size on the gel. And this one you can also see here in this gel. So what we did, we linked now polyethylene glycol to the particles. The more pack you have on the surface of your particles, the bigger it becomes. You can see you get, again, this discrete bands on the gel. And of course, if you have a pack of higher molecular weight, you get a bigger separation on the gel. So our lowest pack, what we can use is 5 kilodalton. And now what we can do, we can prepare particles with a precisely controlled number of antibodies, because we can first make the fractionation of pack, and then we can link the antibodies to the free end of the pack. So how we tested it, we took a tumor model for HER2, and we made now particles with exactly 1 antibody per particle or 2 antibodies per particle with trastuzumab. That's an antibody what binds to the HER2 receptor, and one non-specific antibody that's IgG. Now, these were injected in the group of Davide Prosperi in a mouse model, and with this one we can test now, is it better, for example, to have particles with 2 antibodies or particles with 1 antibody? So if you go to an in vitro model just with a cell culture, if you have particles with 2 antibody, they accumulate better inside the cells than if you have particles with just 1 antibody. But surprisingly, in the in vivo model, we got something different. So if you look here, this one is basically the amount of particles you can find in the tumor. This one here is a particle with 1 trastuzumab, and this one is a particle with 1 IgG. So this part here goes in via this EPR, in fact, enhanced permeation and retention. And so this difference here, that would be the active targeting. So the difference between the non-specific and the specific antibody, because you need to have the non-specific as control to control about size. Now, if we go here to this model where you have 2 instead of 1 antibodies, you can see there's almost no contribution of this active targeting anymore. That means we have a better targeting if we have just 1 antibody instead of 2 antibodies per particle. So how to explain it? We first thought it's a matter of size. But then I learned from a colleague in Mainz that they made actually quite similar experiments. And now our hypothesis is the following. If you have antibody attached to your particle, this particle is recognized by the immune system. If you have more antibody, then it's basically more recognized by the immune system. That means what we speculate, if we have these 2 antibodies per particle, they are just cleared faster by the immune system from the blood. That means they have a lower circulation time in blood. That means they can go less inside the tumor. If you just have 1 antibody, they have a longer circulation time, and in this case, they have longer time to find the tumor, and then there's also a clear effect of this active targeting. Good. Just one thing. I'm almost done. The same thing, measuring size, you also can quantify for binding of proteins. So what you can do, you add proteins, and for each protein concentration, you measure the size of your protein particle complex, and you can see here, you get these nice graphs. So we can see for these model nanoparticles, we really get protein monolayers, and from these curves, we can determine, for example, the binding strengths, how these proteins bind to the particle. So this one we did together with a group of Uli Nienhaus for a lot of particles in test tube. But now, of course, the question is, if we want to characterize what's the protein corona doing in vivo, we would need to have some analysis in vivo. So these data were made by fluorescence correlation spectroscopy. So you measure diffusion constants with an optical technique, and from these you can determine the hydrodynamic diameter. But this one, of course, wouldn't work in vivo. So what we thought now, why do we measure the diffusion constant with an optical method? Because in principle, you also could do it with magnetic resonance imaging. So with magnetic resonance imaging, there are sequences, for example, DOSI, with which you also can measure diffusion coefficients. So we had here now particles with a fluoride label, and this fluoride label provides contrast for magnetic resonance imaging. You can, again, I don't have time to explain the details, measure the diffusion constants. From the diffusion constants, you can get the hydrodynamic diameter. And you can see here, it's pretty consistent with what we did so far with optical techniques. If you add these particles, for example, in human serum albumin or plasma, you get an increase in size. But this one is now a technology what doesn't depend anymore from, let's say, light scattering by tissue. So with magnetic radiation, you don't have any problems with penetrating into tissue, and that's what we are currently trying to do. We're trying to measure protein corona adsorption in vivo with magnetic resonance imaging. And again, the idea is if you have more protein on your particle, it would become bigger. You also can probe degradation. If your particle is degraded, then it would become smaller, and so on. Thank you very much for your attention.
Questions?
You use electrophoresis and the size to separate the particles with, for example, different amount of pace.
Yes.
I guess that there is a limit of how small or large particles you can use for that purpose. So the first example you showed was very small particles. Can you comment about how large particles you can use with this technique?
Yes, so this one is a very good question, and you're absolutely right. So basically, there's a severe limit on size. Let's say you can see just if you run plain gold particles. So we're typically using agarose gels, 2% or 1%. And let's say, if the overall size is, let's say, higher than 30 or 40 nanometers, the amount of separation is too low. So we couldn't see it anymore. So it works, of course, the best, ideally, very small particle, very big ligand. So our standard system is something in the range of the particle with, say, let's say, 5 to 30 nanometer. And then the minimum amount what we have to add as ligand, let's say 5 kilodalton of the pack. So just, for example, with a smaller molecule alone, we wouldn't be able to separate. So what we have to do then, we have to link this molecule to the free end of the pack.
Yeah, you mentioned that with the MRI you can get into in vivo experiments, but what you show here is in vivo experiment or it is in serum? Because what I know from nuclear magnetic resonance is much less sensitive than fluorescence. So you will need very high doses, I guess, to get MRI contrast.
Yes, so I mean, these experiments, I mean, they were not done in vivo. That's what we are trying to do actually right now. You're also correct. I mean, there is basically a sensitivity problem. So what the good thing is, if you have your particle surface accessible, you can put a lot of ligands. So in this case, we are working with fluorine, and this one is not abundant in our body. And so we made some calculations that basically we at the limit, we will not be able to, let's say, make a picture every second, but we estimated, so we would be fine, let's say, with getting one snapshot every 5 minutes accumulation time.
Thank you. One more question.
For your magnetic nanoparticle that used amphiphilic copolymer, how did you add your ligands before coating or after coating in water?
Yes. So this one, in principle, we are free on how to do it. So we can, so basically, I mean, the polymer is formed in organic solvent. So we can add an organic ligand, what's soluble, for example, in chloroform, directly when we synthesize the polymer, and then we add this ligand together with the polymer around the particle. We also can do it different. We can first add the polymer, then everything is water soluble, and then we can use standard EDC chemistry to link a water soluble ligand to the particle. And of course, it plays a role. So if you make the functionalization first in the organic phase, then the ligand, which is hydrophobic, is more inside the polymer shell, whereas if you're making the functionalization afterwards, so when you have everything in water, then the ligand is more on the outside. So for example, for this fluorine labeling, this one plays a role because of course fluorine is highly repulsive, so that would be not good to have it on the surface. So in this case, we first make the ligand modification to the polymer, then wrap the polymer around the particle, and in this way there's basically not so much of the ligand really on the particle surface.
Thank you. Discussion can continue in the...